I'm Jennifer Zaidi, a high school student from Saudi Arabia. Now, because I'm Saudi, growing up in a meat-loving culture, I've always been fascinated by food and its impact on our health. So, my project is entitled Quantification of Choline in popular meat varieties consumed here in Saudi Arabia. So, my curiosity journey has led me to uncover something intriguing, a vital nutrient that plays a leading role in body function, including metabolism, brain development, and muscle movement, called choline. Now, choline could be found in also different kinds of food. It could be found in eggs, meats, and dairy. But the aim of our study is to determine choline concentration in different types of meat, especially those that are consumed in Saudi Arabia. Why? To revolutionize our eating habits and boost public health. So what we've done is we brought different kinds of meats, cooked and raw, homogenized, and used acid hydrolysis extraction. Then we prepared the samples, got them into the mass spectrometry, injected them into the mass spectrometry, which is a very powerful device we're working on. And the mass spectrometry uses a method called selected reaction monitoring, or SRM method. Now, this method is a precise lab technique used to detect and measure specific molecules, like proteins or even smaller molecules, in a sample. It works by first isolating a target molecule, breaking it into smaller pieces, and then tracking unique fragments to confirm its presence. This two-step filtering reduces interferences. Interferences are the molecules or particles that we don't need in our study, making it highly accurate even in a complex mixture like meat samples. And it's ideal for quantifying tiny amounts of substance. Think of it as a molecular search and confirm method. Then we inserted the results into the multi-quant software and got the data analyzed. As we take a deeper look at this figure, it shows the choline standard peaks. And this is the calibration curve we got from the multi-quant. We can see that it shows great linearity, which will help quantify the choline levels in meat samples. Now, here's an example of the peaks that appears using the mass spectrometry. We applied statistical analysis using ANOVA, and as you can see, the results show that choline levels in the camel show a significant higher difference than the rest of the meat samples. Another proof of the significant difference of the camel and the rest of the meat is that the ANOVA p-value equals 0.04, I think. So it is less than 0.5, which means there is a significant difference. And the other figure we're using is the t-test. We use this t-test to assess the effect of cooking in the choline level. And as you can see, the choline level is significantly decreasing after being cooked. Now, why did we pick camel meat to show the difference between it being raw and cooked? Because it's worthy, since it's the highest concentration level. So, our study demonstrates that the elevated choline levels in camel presents a promising natural alternative to the synthetic supplements that are commonly found in pharmacies. Now, to wrap it all up, we've discovered that red meats show higher level of choline compared to white meats. But when red meats are cooked, it shows an increase in choline levels. But white meats show an instant drop of choline levels after being cooked. Now, these findings help us make a healthier, better, and smarter dietary choices. Thank you.